Hello, and welcome to ASGCT's COVID-19 Symposium. I'm Steve Russell. I'm the current president of the Society, and this meeting is going to run for 2 full days through today and tomorrow. We have an outstanding lineup of speakers covering all aspects of COVID-19, from basic virology and epidemiology through pathogenesis, immune response, diagnosis, therapy, and prevention with vaccines. So I guarantee that by the end of this meeting, you'll all know a lot more than you know now about COVID-19. Many of you have submitted abstracts, which have generally been of a very high standard. We had a tough job of selecting from them for oral versus poster presentations. The upshot of that is that we have a great lineup of short oral presentations tomorrow and a great lineup of topical poster presentations, which you can access anytime. All attendees are going to receive an email from ASGCT with a link that will give you a direct access to the poster site. If you just registered recently or you did not receive the email, there's a link on the Symposium site with instructions on how to access the posters. The full text of all abstracts is also located on the Symposium website. So I'd like to thank our organizing committee for pulling together an outstanding program in record time, for vetting all the submitted abstracts, and for agreeing to serve as session chairs throughout the symposium. So you'll be seeing more of the various members of the organizing committee through the meeting. I want to thank our sponsors for bankrolling this meeting. That includes Access Biologicals, Akuos, Cobra Biologicals, Fujifilm, Pendotech, and the 7 companies supporting our sponsored lunch sessions, which I strongly encourage you to register for and attend. They all address COVID-19 relevant tools and technologies. They include Imanos Life Sciences and Viriad on a scalable antibody test for measuring virus neutralizing antibodies, Miltenyi Biotech on the purification of cells from infected patients, Precision Nanosystems on RNA technology underpinning RNA vaccines, Lovelace Biomedical on preclinical animal models, IDT on oligos and probes for qPCR, CRISPR, and NGS, GeneScript on the generation and characterization of anti-spike antibodies, and tomorrow lunchtime will be Corning talking about 3D cell culture for airway organoids and air-liquid interfaces. I also would like to thank the ASGCT staff who've worked and are continuing to work right now tirelessly to make this meeting a success. And we look forward to getting your feedback on what you think of it at the end. So we're going to start the program now with a keynote address from Dr. Anthony Fauci. He regrets he's not able to be here in person, but he pre-recorded the address for us on Friday last week. As you know, Dr. Fauci is the director of the National Institute for Allergy and Infectious Diseases, and he oversees an extensive research portfolio devoted to preventing, diagnosing, and treating infectious and immune-mediated diseases. He's led the federal efforts to combat diseases caused by emerging viruses, including HIV/AIDS, SARS, MERS, Ebola, and now COVID-19. So the title of his presentation is COVID-19, Public Health and Scientific Challenges. So let's put on Dr. Fauci's presentation, please.
Greetings, ladies and gentlemen. I want to thank the organizers for giving me the opportunity to make this presentation before the American Society of Gene and Cellular Therapy. As shown on this first slide, I will be discussing with you today the public health and scientific challenges associated with the historic pandemic of COVID-19. Before I start, I just need to mention that I have no financial disclosures to make. At the end of December of 2019 and the first week of January of 2020, the Chinese identified a new strain of coronavirus, which was the source of an outbreak of pneumonia among more than 20 people identified in the Wuhan section of China. The virus in question was a coronavirus, and the phylogenetic tree, as shown on this slide, indicates the place on that tree where this new virus, which is shown in the yellow colored box, fits next to the original SARS, or the severe acute respiratory syndrome, which was identified as a pandemic coronavirus in 2002, accounting for 8,000 cases and almost 800 deaths. In addition, 10 years later, in 2012, there was the emergence of another potentially pandemic coronavirus called the Middle East Respiratory Syndrome, which emerged in Saudi Arabia and is still smoldering at a very low level in the Middle East. Right now, the disease that's caused by this virus is called COVID-19 for coronavirus 2019. And as mentioned a moment ago, the novel coronavirus that causes this disease is referred to as SARS coronavirus 2. If you look at the global picture, right now, this pandemic has exploded upon the world in a historic fashion, like nothing that we have seen in the last 102 years since the historic outbreak of the 1918 Spanish flu. Right now, there are over 27 million cases in the world with close to 900,000 deaths. The United States has been the most severely hit of all the nations in the world, with the current count being over 6 million reported cases and close to 190,000 reported deaths. If one looks at the pattern of the disease in the United States and compare it to that in the European Union, you can see the pattern in the European Union in blue, in which it peaked and then came down to a very low baseline, less than 10,000 cases per day. Recently in the European Union, because of the attempts to open up the economy, we've seen some resurgence. But this is really very little compared to the situation, which is most extraordinary, in the United States, in which the peak occurred in March and April, mostly associated with the outbreak in the northeastern part of the country, dominated by the New York City metropolitan area. However, when the virus became under control in that section, it never really went down to a low baseline, hovering around 20,000 cases a day, until attempts were made to open up the economy, particularly in certain southern states, which was not successful because there was a surge of cases, bringing the numbers up to about 70,000 cases a day and approximately 1,000 deaths per day. It has now stabilized somewhat at an unacceptably high level of anywhere between 40 and 50,000 cases per day. If you look at the differences of what happened in the European Union compared to the United States, taking two European countries, Italy and Spain, and show that the changes in mobility over time, namely the people who visited the parks and outdoor spaces, was much, much more diminished in Italy and Spain compared to the United States. The same thing holds true in working in the workplaces, a much greater decline in Italy and Spain compared to the dark line of the United States. And even trips to the grocery store and pharmacy, much more diminished in Italy and Spain compared to the dark green line of the United States. So the epidemiology was driven by the degree to which the different countries shut down. Let's have a word about the virology. As I mentioned, this is a beta coronavirus, an RNA virus, whose receptor is a spike protein, what has been shown the component of the virus that binds to the receptor, the ACE2 receptor, is a spike protein whose conformation has been precisely delineated at the atomic level. The receptor for that receptor binding domain of the spike is the human ACE2 receptor, which is widely distributed in tissues throughout the body, including the upper and lower respiratory tract, the GI tract, and most recently identified in cardiovascular and other tissues. The transmission is one of a classic respiratory-borne virus, with the transmission between individuals in close contact. Respiratory droplets, including a degree of aerosol, has now been recently shown to be responsible for a certain degree of the transmission. Infected surfaces occur, but the degree to which this is involved in transmission remains unclear. In addition, the virus is found in various body fluids. Again, the role in transmission is unknown at this time. And also, certain animals, including domesticated animals, have shown to be infected. Again, it is not clear whether this is a source of human infection. Likely it is not. This is a picture of the spray from a typical cough or sneeze of an individual, showing how these respiratory particles can be sprayed over a considerable distance, as shown on this slide. The risk of transmission varies between the type and duration of exposure. Secondary infections are most common among household contacts and people in healthcare settings, particularly when they are not wearing personal protective equipment, and particularly in closed settings, such as cruise ships, nursing homes, and prisons. There are numerous clusters of cases after social or work gatherings, and this highlights the risk of transmission in close contacts. Some cogent examples of this is the now famous report from the state of Washington in the United States, where a single infected choir participant infected 87% of her colleagues in that choir. In addition, there's been community transmission reported at 2 large family gatherings in Chicago, as well as attendees at church events in Arkansas. Again, talking about the risk in close proximity of people in the same enclosed space. Another important aspect of this disease is the fact that between 40 and 45% of individuals who are infected are without symptoms. This becomes even more compelling because we know that up to 50% of transmissions from one person to another occurs from an asymptomatic person to an uninfected individual. Talk a little bit about prevention. There are personal preventive measures that people have now implemented that clearly are having a positive effect on preventing infection, such as diligent hand washing, avoiding close contact at least 6 feet from individuals, covering the mouth and the nose with a cloth or a classic mask, avoiding face touching, and regular disinfecting surfaces. The public health measures are those that are mandated, such as distancing orders, stay-at-home orders, modification of or canceling schedules at schools and businesses, bans on public gatherings, various entry and exit travel restrictions, as well as identification of cases and contact tracing. The clinical manifestations are protean. We know that the incubation period has a median of approximately 5 days, and the range is 2 to 14 days. The clinical presentations are very similar to that of a flu-like syndrome, with the symptoms and signs shown on this slide. Of note, there is a peculiar loss of smell and taste, which can precede the onset of respiratory symptoms in many individuals. If you look at the relative percentage of mild to moderate disease compared to severe disease, clearly the overwhelming majority of individuals have mild disease which does not require any specific therapeutic or medical intervention. However, about 20% of individuals have severe disease, and the case fatality rate varies from a few percent to up to 25% fatality rate in individuals who require being put on a mechanical ventilator. The manifestations of severe disease vary, predominantly acute respiratory distress syndrome, but also hyperinflammatory syndromes and injury to the heart, to the kidneys, to the neurological system, as well as a hypercoagulable state with microthrombi in small vessels and thrombotic events causing stroke in larger vessels. There is also a very interesting multi-system inflammatory syndrome in children resembling classic Kawasaki syndrome. The people who are at increased risk for severe COVID disease are older individuals, and as shown in this slide, if you look at the cases of hospitalization per 100,000 population, you can see the extraordinary difference on the left-hand side of younger individuals with a low rate of hospitalization compared to the profoundly large number of individuals per 100,000 who are in the elderly group from 75 to more than 85 years. In addition, people of any age with certain underlying medical conditions, as shown on this slide, have a strong risk of severe disease. These underlying conditions include serious heart conditions, including cardiomyopathies and coronary artery disease, chronic renal disease, chronic obstructive pulmonary disease, diabetes. Obesity is quite important, and immunosuppressed states such as associated with cancer and solid organ transplant. There are some medical conditions that may have a risk that's increased, but not as severe as the ones I just showed you. These include those that are shown on this slide. I particularly want to point out hypertension as well as chronic lung disease. It is important to point out that there are substantial disparities in racial and ethnic groups, with African Americans, Latinx, as well as Native Americans and Alaskan Natives, to having a higher degree of infection as well as complications that lead to severe outcomes. If you look again at the rate per 100,000 population of hospitalization, look at the top bar where there are 333 per 100,000 blacks who are hospitalized compared to only 71 whites per 100,000. With regard to diagnostics, there are molecular tests to detect the actual virus. These are highly sensitive and specific. They take longer to do, are more expensive, but highly accurate. Antigen tests can be scaled up much, much greater. They don't detect the virus. They detect proteins associated with the virus. And then there are antibody tests to determine if a person has actually been infected. An important breakthrough has occurred most recently where the COVID-19 card test by Abbott, which is a really quite a milestone in that it is a highly sensitive and specific, inexpensive, and you get the result in 15 minutes. It requires no specialized lab equipment and is in the form of a credit card-sized apparatus. This is something that has now been purchased in large amounts by the federal government and will be used in nursing homes, prisons, schools, and other individual cases that required broad surveillance. Moving on to therapeutics, the United States National Institutes of Health has formed a guideline panel, which is really a living document that's online, giving real-time updates on clinical data from clinical trials that have been published as well as those that are in pre-publication to give the clinicians an up-to-date, real-time capability of knowing what the best clinical practices are. It's online available at COVID-19 treatment guidelines.nih.gov as shown on the bottom of this slide. I'll get to two drugs that have already been shown and recommended by the guidelines panel to be used for clinical practice, but there are other examples of drugs that are under investigation in clinical trials, including antivirals, convalescent plasma, hyperimmune globulin. Monoclonal antibodies are now quite promising, as well as inhibitors of cytokines, such as other immunomodulators. And then there's the anticoagulation that is being used now for the hypercoagulable state. The first of the two randomized placebo-controlled trials that showed efficacy of an intervention was the use of remdesivir, which is a direct antiviral, used in a randomized controlled trial of over 1,000 individuals in countries throughout the world. It showed a clear-cut benefit in decreasing the time to recovery, highly significant, but a modest decrease by 32%. This is in individuals who are hospitalized with indication of lung involvement. A study from the UK, again, a randomized placebo-controlled trial involving thousands of individuals, which was looking at individuals who are hospitalized on ventilators or requiring oxygen. And the study showed a significant diminution in mortality in individuals receiving dexamethasone compared to those who are on the placebo. Other subsequent studies, again, confirmed the role of corticosteroids in decreasing 28-day mortality by a significant percentage. And finally, vaccines. We in the United States have taken a strategic approach of harmonizing up to 6 or 7 clinical trials using various candidates. By harmonization, we mean a common data and safety monitoring board, common agreed upon primary and secondary endpoints, and common immunological parameters to be followed. This is a list of the different platforms. There are 3 major platforms involving 7 individual candidates. The nucleic acid platform using messenger RNA, the viral vectors using either a chimp adenovirus, a human adenovirus, a VSV, or a measles vector, and then the typical classical protein subunit. As shown on the right-hand part of the slide, 3 of these trials are already in phase 3 advanced testing for efficacy and safety. It is projected that we will get an answer by the end of 2020, generally felt to be about November or December, or possibly the beginning of 2021. We could get an answer sooner. That is unlikely, but not impossible. We project, because of favorable data in the animal model and in the phase 1 clinical trials, which showed the induction of a very good neutralizing antibody response, making us cautiously optimistic. Although you can never guarantee anything with vaccines, we're at least cautiously optimistic that we will have an answer by the end of 2020 and we'll be able to start distributing vaccine in a meaningful way in 2021. I want to close by putting up this slide, which is an indication of a COVID-19 prevention network where one can go online and examine what is available in clinical trials and even volunteer, if you would like, for one of the clinical trials, particularly the vaccine, by merely clicking on to coronaviruspreventionnetwork.org, one word, coronaviruspreventionnetwork.org. And again, thank you for giving me the opportunity to make this presentation.